Dear students, today I would like to teach you on classification of drugs. Classification of drugs, how we can classify drugs, and on what basis we can classify drugs. So it is very important to understand classification of drugs. So we can classify drugs on the basis of different factors. Okay, so first factor, we can classify drugs based on the basis of pharmacological effect. Pharmacological effect. Pharmacological effect. The first classification, we can classify drugs on the basis of pharmacological effect. What is pharmacological effect? Pharmacological effect is nothing but a biological effect. Okay, suppose a patient suffering from pain, okay, and that patient can take aspirin as a drug. After taking the drug aspirin, after some time he will get relief from that pain. Okay, so these biological effects, what I said in this, that pain and relief from pain, these pharmacological effect. So we, on the basis of pharmacological effect, we can classify drugs into different classes. Example, so analgesic, okay, analgesic, analgesics, okay, analgesics. The example for analgesics is aspirin. Okay, so when you take aspirin, okay, that gives you relief from the pain, right? So therefore we can classify drugs on the basis of pharmacological effect. Suppose I'll tell you another thing, anti-inflammatory drugs. These kind of drugs can cure the inflammation in the cells, right? Or infected cells can be cured by these kind of drugs, anti-inflammatory drugs, right? Like this, or anti-cancer drugs, okay, anti-cancer drugs. Anti-cancer drugs also, you know that these kind of drugs can used to cure the cancer, or these kind of drugs can be used for the treatment of cancer disease. Okay, so the based on the pharmacological effect, so we can classify drugs into different categories like analgesics, anti-inflammatory drugs, anti-cancer drugs, anti-HIV drugs, okay, like this, etc. Okay, so this is the way we can classify drugs on the basis of pharmacological effect. The second one is, we can classify drugs on the basis of drug action. On the basis of drug action, okay. The second criteria to classify drugs into different classes on the basis of drug action, right? Suppose what is the drug action? Drug action, you know that each drug has its own way of generating response called drug action. Okay, each drug its own way of generating a response called drug action. Suppose, example I will give you, hypertension drugs. Okay, hypertension, hypertension uh, drugs. There are many number of hypertension drugs are available in the market, but all of them has its own pathway of action. So finally they can work on the hypertension problem. Okay, so drugs, there are many drugs, but different pathways. Okay, different pathways. Different pathways is nothing but different mechanism of action. Okay, mechanism of action is different in these drugs. Okay, so we can classify drugs on the basis of drug mechanism of action. So this is the second classification. So mainly depends upon the drug mechanism of action. So can I give you one example on this? So alpha glucosidase inhibitors. Okay, alpha glucosidase inhibitors. So that means these kind of drugs can cure can be used in the treatment of type 2 diabetes through this mechanism of action. These kind of drugs can interact with this enzyme alpha glucosidase. Okay, these kind of drugs can inhibit the function of alpha glucosidase enzyme. So that's how this is the mechanism of type 2 diabetes. One of the type 2 diabetes drug, alpha glucosidase inhibitor. So alpha glucosidase enzyme function can be inhibited by these kind of drugs. So the mechanism of these drugs through inhibition of alpha glucosidase enzyme. So I hope you will understand what I want to say. Drug action. Based on the drug action, we can categorize or classify drugs into different categories. Okay, so it is important to understand that on what basis, on what criteria we can classify drugs into different categories. Okay, suppose topoisomerase inhibitors. Topoisomerase inhibitors. That means these kind of drugs inhibit the function of topoisomerase enzyme. Okay, so that is the mechanism of action. Okay, these drugs enter into the body and interact or interact with the topoisomerase enzyme and stop the function of topoisomerase enzyme or alter the function of topoisomerase enzyme. So that's why the mechanism of these kind of drugs is like this. So therefore the second classification of drugs on the basis of drug action. What about first criteria I said? What is the first criteria I said? Classification of drugs on the basis of pharmacological effects. Okay, and the second criteria to classify drugs into different categories is drug action. Okay, so say for example there are enough number of drugs which can treat hypertension, but all of them are not working in the same pathway. they have its own their pathways. Okay, so the here it is very important to understand the pathway or the mechanism of action. So on the basis of mechanism of action or drug action, we can classify drugs into different categories. Say for example, alpha glucosidase inhibitors, topoisomerase inhibitors. Okay, so this is the way we can classify drugs into different categories. The third classification of drug on the basis of chemical structure. Chemical structure. So here it is very important is basic skeleton structure. Basic skeleton structure. Example, sulfonamides. Sulfonamides. Okay, sulfonamides means all these kind of drugs or these class of drugs have same basic skeleton structure. Sulfonamides. Okay, they have similar basic skeleton structure, chemical structure. On the basis of chemical structure, we can classify drugs into different categories. Sulfonamides, like what I can give some more examples. Benzodiazepines. Benzodiazepines. So these kind of drugs all comes under this classification, have basic skeleton of benzodiazepine structure, chemical structure. Okay, I hope you will understand about this classification. Okay, sulfonamides, benzodiazepines. Sulfonamides and benzodiazepines. Okay, so this is the third classification of drugs. The next one is the fourth classification is based on targeting molecule. The fourth classification of drugs is on the basis of targeting molecule or target molecules. Okay, some drugs can target metabolic process. I'll give you some examples. Drugs acts on the metabolic process. Next, drugs acts on the genetic material. Okay, or drugs acts on the ion channels. Okay, so here some more examples. Drugs acts on the cell wall. Here you observe metabolic process, genetic material, ion channels and cell walls. These drugs, all drugs comes under this category, will target metabolic process. Okay, so these drugs acts on the genetic material. All drugs which comes under this category will target genetic material. Okay, like ion channels, like cell wall. Okay, so these are all target molecules, right? These are all target molecules. Based on the targets, we can classify drugs into different categories. So these are the different classification of drugs. I hope you will understand. What about first one? On the basis of pharmacological effect. Pharmacological effect. What about second classification? On what basis? On basis of drug action or drugs pathway or mechanism of action. And what about third one? Third classification, on the basis of chemical structure. On the basis of chemical structure. What about the last one? Drugs classified are based on the target molecules. Target molecule or target macromolecules. We can say that molecular targets are target macromolecular targets. You can, this is the correction, you can make it macromolecular targets. Okay, so these are the 4 criterias by which we can classify drugs into different classification. Pharmacological effect, we can classify drugs as analgesics, anti-inflammatory, anti-cancer agents, anti-HIV agents, anti-diabetic agents, okay, and antibiotics. All these class of drugs which can classified based on the pharmacological effect. The second one, drug action or pathway or mechanism of action. Okay, so in this you have seen some examples: alpha glucosidase inhibitors, okay, topoisomerase inhibitors. Okay, so that is the mechanism by which drug can act and gives or produces biological response. The third one is chemical structure. On the basis of chemical structure, so we can also classify drugs into different categories like sulfonamides, benzodiazepines. Okay, so in this we can classify on the basis of the basic skeleton structure or chemical structure. So which are similar, they can comes under this category. And the final one, on the basis of macromolecular targets. Okay, so the drug which target the macromolecular part, on the basis of that we can classify drugs based on the molecular targets, right? The last one is very very important. This is the latest one. Okay, this gives a lot of information, or this helps a lot of this helps to clinical trials a lot, because in the clinical trials, if they know which target molecule they target, it is very easy for them to do clinical trials. So therefore, so now we are going to see some drugs in this unit. First, drugs acting on metabolic processes and cell wall. Okay, and also drugs acting on genetic material, DNA, RNA, okay, and carbohydrates, enzymes, receptors, transport proteins, okay, and lipids. All these are macromolecular targets. Okay, so if a drug targets which molecular target, macromolecular target, on the basis of that we can classify drugs into different categories. So these are the 4 ways we can classify drugs into different classes. So next I'll explain you about uh, enzyme inhibition. Enzyme inhibition and its types. Enzyme inhibition and its types. So you already know about enzymes. Enzymes are biological catalysts. They can reduce the activation energy so that excels the rate of the reaction of biochemical reactions which are occurring inside our body. Okay, enzymes are very stereospecific, very specific in their nature. Okay, so they will produce 100% extraordinary exclusive products. Okay, so they are very, very stereospecific reactions. Okay, enzymes are biological catalysts. First of all, enzymes are made up with, they are nothing but proteins, right? They are nothing but proteins. Okay, their structure, chemical structure, you can understand. If it is protein, that means enzymes are made up with amino acids, monomers, right? So they can act as biological catalyst, and they are decreasing activation energy accelerate the rate of the reaction accelerate the rate of the reaction, and finally involves in the mechanism of reaction. After completion of the reaction, returns to its original state. Returns to its original state. Okay. Enzymes are made up with amino acid units, monomer units. They can link together through peptide bonds. So they have primary, secondary, tertiary and quaternary structures, and they have they are biological catalysts. That means they act as a catalyst. They participate in the reactions without changing itself. It does involve in the mechanism of the reaction, and it decreases the activation energy. Okay, it decreases the activation energy in that reaction so that accelerate the rate of the reaction. Okay, and finally returns to its original state, and again it participates in the same function again and again repeatedly. So that is the beauty of enzymes, right? So we'll try to learn about enzymes more clearly. How it participates in the, how it acts as a catalyst in biochemical reactions. So without enzyme, with enzyme, so there is a huge gap in the rate of the reaction. Okay, there is a huge gap with enzyme as a catalyst in that reaction, without enzyme in that reaction. Okay, so the rate of the reaction is, the difference in the rate of the reaction is so high. So that is the um, great function of enzyme. So uh, how it reacts in the how it participate in the biochemical reaction as a catalyst. I would like to explain you. Okay. So you know that enzymes are tubular proteins, right? Suppose this is the structure of an enzyme. Okay, it has a deep cavity. It is a cavity or a hole. Okay, so this is called active site of an enzyme. Active site of enzyme. All the reactions takes place in this active site. Therefore, active site is very important in enzyme. Okay. So, what is active site of an enzyme? Active site, a deep cavity from surface of the enzyme to the middle of the enzyme, or in a deeper. So a hole or a hollow, a cavity will be formed, that is called active site of an enzyme. Okay. So what how reactions takes place in this active site, that I want to explain you. In this active site, what happens? Amino acid functional groups, amino acid residual functional groups exist in the active site. Okay. In the active site surface, amino acid residual functional group exist. Those functional group interacts with the substrates. Substrates are nothing but starting materials in enzymatic reactions, right? Okay. So suppose you know about amino acid structures. In amino acid structures, you have hydroxyl groups, carboxylic groups, right? And aromatic rings and aliphatic skeletons and sulfur NH2 groups, right? All these are functional groups which can mm, exist in active site of an enzyme. Okay, so these amino acid residues or functional groups interact with the substrate. So only interactions takes place. Enzymes does not participate in the reaction. Okay, so it only interactions, it takes place interactions. Okay, like hydrogen bonding interactions. Okay, electrostatic interactions, van der Waals interactions, and what do you say, uh, hydrophobic interactions. All these kinds of interactions takes place between this active site and substrate, so that the electron transfers takes place. Okay, the electron transfer and hydrogen transfer. Okay, these kind of transfers takes place after completion of the reaction. After completion of the reaction, enzymes will return its original state. Okay, but substrate will converted into product and releases. Okay. Suppose, so this is the rate of the reaction, rate of the reaction, and this is the uh, uh, reaction progress. Okay, suppose you take is the starting material. So if this starting material convert into product, so this is the product, this is the starting material. Okay, so this is called activation energy, the gap between uh, this and the this is the activation energy. Okay, so this can be activation energy. This is the transition state. Okay, transition state. Okay, so starting materials, first it converted into transition state and finally converted to product. Okay, so if the starting material converted to product, it needs this activation energy, this kind of activation energy. Suppose this is without enzyme. Suppose with enzyme the activation energy decreases. Okay, so this is without enzyme. That reaction, this activation energy is with presence of enzyme. With the presence of enzyme. Okay, so here you can see, so this is the on y-axis rate of the reaction, the x-axis reaction progress. Okay, suppose the starting material has zero energy at this level, and if you give some energy, it will first reach to the transition state and finally converted to as a product. Okay, and the required energy from starting material to transition state is called activation energy. So without enzyme the activation energy is more. Okay, so with the presence of enzyme the activation energy is less.